You're probably wondering, what is a mutation? Well, genetic mutations are changes to your DNA sequence that happen during cell division when your cell makes copies of themselves. Your DNA tells your body how to form and function. Genetic mutations could lead to genetic conditions like cancer, or they could help humans better adapt to their environment over time. It affects the phenotype of the eye color. It should not affect anything other than the pigment of the eyes, unless it is acquired heterochromia. Then organisms might experience symptoms like swelling, benign, and malignant tumors of the iris, bleeding in the eye, and eye injury caused by other symptoms. For example, David Bowie's left eye dilated after an injury and resulted in acquired heterochromia.
There are 3 different types of heterochromia: central, complete, and sectoral or segmental. Central heterochromia is when there are 2 different colors in the same iris. The outer ring is a different color from the inner ring and surrounds the black circle of the iris. Sectoral heterochromia causes part of one iris to be a different color than its intended color. And complete heterochromia causes completely different colored eyes. For example, one blue eye and one green eye.
In order for your eyes to have color, your eyes need to have the protein called the P protein. This is the specific protein that produces the eye pigment. OCA2 is what makes the P protein. The OCA2 gene is not present, there is no P protein. The P protein is responsible for maturing the cell structures that produce/store melanin, called melanosomes. It determines the amount, type, of the color present in the iris, meaning brown equals more pigment and blue equals less pigment.
DNA is made from 2 connecting strands. Each strand is made of a backbone. These backbones consist of a sugar called deoxyribose and a phosphate group. Connected to each sugar is a base called a nucleotide. These 2 strands of DNA are connected in the center by corresponding nucleotides. DNA has 4 different types of bases: adenine, thymine, cytosine, and guanine. An enzyme called helicase splits these 2 strands apart from each other and leaves the individual nucleotides unpaired. These nucleotides are specific codes for proteins called base pairs. The codes for DNA are determined by the letters A, T, G, and C. A pairs to T, and C pairs to G, and vice versa. After this split, this is where mRNA, also called the messenger RNA, comes in. mRNA is a type of RNA similar to DNA in that it has a backbone of a sugar and a phosphate, except its sugar is called ribose, and instead of having 2 strands connected to each other like DNA does, it only has 1 strand. It carries its own codons to match with the codons of the original strand of DNA. It changes the code to match A to T, U to A, and G to C. This makes a copy of the message of which proteins to make from the DNA and carries the copy out of the nucleus and into the cytoplasm.
Heterochromia can happen to any living thing at birth from mutation and any age later on in life, from an outlier like damage done to the eye, but it is most common in dogs, specifically Australian Shepherds. This is Gerard, a 3-year-old Australian Shepherd. Heterochromia is a genetic gene that can be passed from parent to offspring, but to his surprise, Gerard's mother didn't have heterochromia. This doesn't mean there is something wrong with him, this just means that his mom didn't have the recessive gene, or his cells just happen to mutate because of other factors. In most cases, it is a genetic mutation, a benign mutation affecting the development of the melanin in the iris. Gerard has 1 blue and 1 brown eye, which is the most common combination of colors within heterochromia. The only genetic factors known to produce blue eyes are 2 cases associated with coat coloration. Genetic mochicism, aka mutations, occurs when genetic recombination, or a mutation occurs during mitosis, then resulting in a genetically different cells.
According to a research document posted to the National Library of Medicine, melanocyte cells produce melanin that is deposited in the melanosomes. Variation in the number of melanosomes and the amount of pigment in each melanosome present determines the eye color. The type of pigment affects eye color as well. Both groups of pigment form from the L-tyrosine that is converted into L-dopaquinine by trinase. Trinase is related to protein 1 and converts L-dopaquinine to melanin, while the addition of cytosine forms pheomelanin.
2 major genes responsible for eye color are HERC2 and OCA2. During the first studies to classify genes for eye color, OCA2 was believed to be the dominating factor for eye color determination. After more recent studies, HERC2 was found to be equally important to determining eye color. RNA is used in this process sort of like how ingredients are necessary to make a cake. Every part is needed with the exact measurements, or else there won't be a cake.
Mutations like heterochromia can be caused when the RNA makes the wrong measurements for the cake, or in Gerard's case, the P protein. For example, in the process of transcription, the mRNA could accidentally pair the wrong two nucleotide bases together, causing the wrong type of protein to be created, or possibly skipping over the creation of a protein altogether. Back to the P protein, it is one of the main proteins responsible for heterochromia. tRNA, or transfer RNA, takes the codes from the codons and uses them to assemble a protein. They do this by adding one amino acid at a time. Codons, as mentioned before, are the instructions. They are a 3-letter sequence from the mRNA. These code for a specific amino acid to be added to a protein chain. It also sends a stop or start signal for a protein synthesis. These instructions come from the translated copy of the DNA to RNA.
Translation, the second step in getting from a gene to a protein, takes place in the cytoplasm. The mRNA interacts with a specialized complex called a ribosome, which reads the sequence of the mRNA nucleotides. Each sequence of 3 nucleotides, called a codon, usually codes for one particular amino acid. Amino acids are the building blocks of proteins. A type of RNA, called transfer RNA, tRNA, assembles the protein, one amino acid at a time. Protein assembly continues until the ribosome encounters a stop codon, a sequence of 3 nucleotides that does not code for an amino acid.
The 2 missense mutations that are involved in the causing of heterochromia are localized in the N-terminal part of the paired domain, where the nucleotides pair together. This means an amino acid is exchanged for another, and the P protein may not have the correct shape to function, which leads to the difference in coloring of the iris.
According to a research document available on Gale, DNA testing of 6,000 dogs, quote, amino acids are the building blocks for protein. When they join together, they create a protein. A string of these conjoined amino acids is a polypeptide chain. These make up proteins. The only genetic factors known to produce blue eyes are 2 cases associated with coat coloration, Merle and Piebald, dogs that have patchy coat colors due to mutations in pre-melanosome protein, PMEL17, and melanogenesis-associated transcription factor, MTF. That can lead to 1 or 2 blue eyes, or slices of sectoral heterochromia, when depigmented regions extend across the face. PMEL is regulated by MITF, the master regulator of melanocyte development. Rarely, non-mural, Australian Shepherds have unexplained cases of solid blue eyes or complete heterochromia, as in huskies, and the genetic basis of this trait is similarly unknown, end quote. So you see, Gerard has nothing wrong with him. Heterochromia is something out of his control, and he is still just as beautiful.